Good evening, everyone, and welcome to today's session. We'll start in 5 minutes so that more people can join in. Okay? So we're gonna start with our discussion, but let's see where we left in our last class. So we are basically discussing about the secondary structures in protein and major secondary structures which are present in protein, and why is there limited number of secondary structures which are present when there are infinite number of possibilities, right? So we digged around Ramachandran plot to find the answer for this question, and then we looked at how the structures are being determined, and then we had some discussion related to alpha helix, which is a secondary structure. What are its properties to define whether this is a secondary structure or alpha helix, right? And then we started our discussion for beta sheets, which is another type of secondary structure. So there are two different types of beta sheets, the parallel beta sheet and anti-parallel beta sheet. You can see the hydrogen bond arrangement in anti-parallel beta sheet and parallel beta sheet. Then we saw some basic information regarding this beta sheet itself, so I'm gonna recap a little bit for you guys so that we can have a revision simultaneously. So let me share the screen. Yeah, 1 minute. I'll just quickly go to my screen. I hope you guys can see it. This is a very brief about
Friends, let's begin with some example. This is myoglobin. This protein, as you see, it is made up of 8 alpha helices. In the case of beta pleated sheet, this is silk fibroin, that is predominantly made up of beta sheets that is joined by loops. Majority of proteins contain both alpha helices and beta sheets. So this is amylase. As you see, these are the beta sheets and these are the alpha helices that is present in amylase. Now let us see what are secondary structures before moving into the difference of alpha helix and beta sheets. A primary structure is a linear chain of amino acid that is joined by peptide bond. So this is the primary structure. This primary structure undergoes regular repeated folding and forming the secondary structure. And this is primarily formed by hydrogen bonding. The helical conformation is called the alpha helix and an extended conformation is called the beta pleated sheet. These beta sheets are made up of beta strands and alpha helix is the helical conformation of a polypeptide chain. Now let us see the difference between alpha helix and beta sheets. Alpha helix, it is a spiral configuration of polypeptide chain that is formed by hydrogen bonds. As you see, these are the hydrogen bonds involved. Right-handed helices are more common, whereas beta sheets, 2 adjacent polypeptide strands. These are called as beta strands that are bonded by hydrogen bond. So these 2 or more strands may interact together to form an extended sheet-like structure called the beta sheet. Now let us see the bonding in alpha helix. In alpha helix, as you see, the bonding is between this carbonyl oxygen and amino hydrogen. This is the bonding. So as a result of this bonding, the structure becomes helical as the amino acids are far away. So this is the second bonding. Carbonyl oxygen and amino nitrogen combines by hydrogen bonding, making the structure helical. Whereas in beta sheets, so this is the first peptide strand and this is the second peptide strand. So hydrogen bonding is between the amino hydrogen of one strand and carbonyl oxygen in the backbone of the second strand or adjacent strand, as you see. So these are some of the bonds. So here also you can see. The third difference is in alpha helix there is intramolecular hydrogen bonding. The bonding is within the polypeptide chain. As you see, all the bonding are present within the polypeptide chain. A single polypeptide chain is involved. Whereas in beta pleated sheet, there is intermolecular hydrogen bonding. There are separate peptide strands or polypeptide chains that forms a fully extended linear sheet-like conformation. So the bonding is intermolecular or between peptide strands. Alpha helix can be a single chain like this, it's a single chain and is of only one type. Whereas beta sheets, there are two types of beta sheets, depending on the orientation of the peptide strands involved. So first one is a parallel beta sheets. As you see, this is the N terminal and this is a C terminal. This is the first peptide strand and this is the second peptide strand. If both these peptide strands runs in same direction, this is NC direction, this is amino terminal to carboxy terminal direction. If both runs in same direction, it is called as parallel. Then this is the anti-parallel strand. As you see, this first strand runs in N terminal to C terminal direction and this second strand runs in opposite direction, that is N terminal to C terminal. In opposite direction, therefore called as anti-parallel strands. Anti-parallel strands are more stable because of more suitable orientation of hydrogen bonding within these strands. Next difference is regarding the dimensions. An average alpha helix is 10 residues. It can reach between 4 to 40 residues. A complete turn is of 3.6 residues. As you see here, this is the turn and that is made up of approximately 3.6 residues. In beta sheets, a beta strand is a polypeptide chain typically of 3 to 10 amino acids long with a backbone of an extended conformation. Now regarding the amino acids involved. The most preferred amino acids in an alpha helix is alanine, leucine, methionine, etc. And the least preferred is proline, then bulky amino acids like tryptophan, tyrosine, glycine, etc. Proline cannot fit into this helix, very difficult to form the bond and introduces a kink, therefore also called as helix terminator. Proline is often called as helix terminator. In the case of beta sheets, it is an extended conformation, so there is enough space for bulky amino acids like tryptophan, phenylalanine, tyrosine, etc. So these amino acids, bulky amino acids, are commonly present in beta pleated sheets. Hope you understand the difference between alpha and beta helices. If you are benefited, please subscribe this channel and share this video.
Right, so that was the basic difference between alpha helix and beta sheet, which are the predominant secondary structures in protein. So if we talk about alpha helix, let me take a pen. Yeah, so in alpha helix, the type of hydrogen bonds are intramolecular, that is, the bonds are within the same polypeptide chain, right? If we talk about beta sheets, right, then they have intermolecular hydrogen bond, meaning that there are different polypeptide chains between which the bonding takes place. Okay, so these are the major differences, and those properties which we studied about, that what keeps the protein stable, like non-covalent interactions. Higher the number of non-covalent interaction, more stable the protein will be. So it applies for both the type of secondary structures, that is alpha helix as well as beta sheets. Okay, so this is the basic properties of beta sheet. Let me beta sheet. So in this, the polypeptide is fully extended because it is not coiling within self, right? Hence it will be the extended version of the polypeptide chain. Then there are 2 residues per turn, unlike alpha helix which had 3.6 residues per turn. Okay, the translation is 3.4 angstrom per residue. These are stable when incorporated into a beta sheet. If we say that why a single beta strand, because if we look at beta sheet, this is one beta strand, this is another beta strand, right? So why we are not considering beta strand as a sole standing secondary structure? They are secondary structures, but it is difficult for a sole beta strand to attain the stability which it will attain when it is paired with other beta strands. Okay, hence standalone beta strands are difficult to find in the physiological condition, but beta sheets are oftenly found in the physiological condition, especially in the proteins. Okay, then hydrogen bond between peptide groups of adjacent strands, which we have discussed that in the beta sheets the hydrogen bonding pattern is intermolecular, that one polypeptide chain will make the hydrogen bond with the another polypeptide chain to come up with a beta sheet as a secondary structure. Okay, then we have adjacent strands which can be parallel or anti-parallel, which we have discussed, right? So, and this is the area in which the beta strands belong. So this is the Ramachandran plot as we have discussed earlier. Okay, so there are parameters for regular secondary structures. So if you remember, when we were discussing about the Ramachandran plot, there are allowed region, there are disallowed region, and there are restricted regions in which a protein might exist if we somehow change the phi and psi values, right? So these are the regular parameters for such secondary structures where they fall in the Ramachandran plot. So if we talk about anti-parallel beta sheet, the phi angle is minus 139 while the phi angle is plus 135. Okay, if we talk about parallel beta sheet, then the phi angle is minus 119 and phi angle is 113. Right-handed alpha helix is minus 57 and minus 47 respectively. 310 helix, as because we have only discussed about the alpha helix because they are the major type of secondary structure, but they are not the only type of helix which are present. There are different types of helix like 310 helix or pi helix. Okay, so in 310 helix, the phi and psi values are ranging from minus 49 and minus 26. If we talk about pi helix, the phi value is minus 57 degrees while phi value is minus 70 degrees. If we talk about polyproline 1, polyproline 2, polyglycine 2, these are a type of extended peptide chains which are found which if you want to locate on the Ramachandran plot, they have phi and psi values as listed here. Okay, so apart from alpha helix, beta strands or beta sheets, there are other secondary structures which make the protein or incorporate the structure in the polypeptide chain to make it as a functional protein, right? So turn S is another type of secondary structure. So basically it's a secondary structure which are connected by loop regions, correct? So we'll see an example in the coming slide. So the lengths vary, shapes are irregular, loop regions are at the surface of the molecule, rich in charged and polar hydrophilic residues. The major role which turn S provide is connecting units, binding sites, enzyme activities, and wherever there is functional role. Then loops are often flexible, adopt different conformations. So beta turns, there are two types, type 1, type 2. You don't need to dig much about it. It is just an introductory class. So what we mean by turn is that if we have beta sheets, right, if we have alpha helices, so a protein will have all the secondary structures simultaneously, right? It can either be predominantly alpha helix or beta sheets, or it can share both the structural components. So loop regions are the ones which are not as structured as these secondary structures, but they do help in connecting the protein from one secondary structure to another secondary structure. So they are very flexible, and as they are loops, they don't have any protection of secondary structure that they can hide their hydrophilic amino acids, so they are often exposed to the cytoplasm. If we talk with respect to the membrane proteins, I might show you a system maybe in the coming days. So basically what happens is you have proteins which are transmembrane proteins, right? So in the membrane region where the protein is residing, it will be predominantly alpha helix, while the regions which are above the leaflet of the membrane, they'll predominantly have loop regions, and they don't have any such structures. So I'll show you one such protein in the coming class with the membrane and with the protein embedded in the membrane, and we'll see if I can get that. We'll see how it is transporting something, like it's a channel and all. So I'll figure it out and I'll show you guys how it basically looks like. So this is a type of protein called thrombin. All of you might have heard its name, and you can see it's a typical protein, right? So you can see helix here, you can see one beta strand here, another beta strand here, another here, another here. So basically this is one beta sheet, this is another beta sheet, this is helix. So all these are connected via these loop regions. So these are turn S, so which connects one secondary structure to another secondary structure. So this is some very basic information about turn S, that if we see how they are connecting the secondary structures, so the number of loop regions and the number of residues in the loop region, because if you are saying it is unstructured, so it can be a simple polypeptide chain, extended polypeptide chain, it will also considered as a loop. But when the experiments were performed, it is observed that generally there are loop regions which have different residues, but the predominant one is 3 to 4 residue per loop. Okay, so you can see the frequency distribution here, that in the number of loops on the x-axis, so this is predominantly present, right? So a loop can be considered as a loop or a turn S when it has 3 to 4 residue. It is predominant, though we have 5 to 6 also, but the predominance is for the 3 to 4 residue long loop. Okay, then again this is the this is the protein sequence of the thrombin, and you can see this is the Ramachandran plot depicting where this protein lies. So it is distributed all over the Ramachandran plot. Why? Because it has beta sheets, it has loops, it has alpha helixes also. Okay, so before moving on to a different topic of protein data bank, that from where we got the sequence, from where we got the structure, let's have some little bit idea about the motif term or the domain term, which will be oftenly using in the coming classes for describing the structure of a protein. Okay, so let me share my screen with you guys. Yeah,
it's Dr. B. This is a part 2 of a video on protein structure. So now that you kind of have a fairly good idea about the generic folding aspects of protein structure, I want to talk a little bit more about something called a domain and kind of what that is. You look at this thing here, this is a protein called P53. It's a tumor suppressor protein, and its job is to basically either correct DNA damage errors or to direct the cell into apoptosis, which basically means blow itself up if it can't fix the issue. And there are some diseases associated with mutations. Li-Fraumeni syndrome is one. There's a bunch of others. But you look at this and you're like, this looks like a bowl of spaghetti. And from the last video, you probably can point out a few things, like maybe a few beta sheets and definitely some alpha helices and whatnot. But you know, let's add some life to this, and that's to me what domains are. And so to kind of get a better sense of them, let's just kind of look at this protein and kind of what it needs to do, and then we'll talk about what a domain is and what its domains are. So this is the P53 tumor suppressor protein, and this is just a smattering of interactions. It's not all of them, but this thing directly connects with and activates some other protein, specifically this one right here called MDM2. You do not need to know any of this. I'm just giving you a sense of how most proteins really work by interacting with other proteins, depending on what the cell needs to do. So now the question is, how in the world does something like this spaghetti looking thing actually do all of this? Well, it has parts of its body that do different things. And just like you and I have, fingers that have a certain way that they look and move and function, that's very similar to domains of proteins. So on that note, let's take a look at the actual domains of the P53 tumor suppressor protein. Now, excuse me, first of all, what the heck is a domain? Generally, it can get complicated, but keeping it simple, it's basically a segment of a protein that can fold on its own and work independently from other parts of the protein. And the structural and evolutionary and functional units of proteins are another term for what a domain would be. And they're also homologous segments in evolution. So it's a great way for us to kind of be able to compare one way different proteins that might all have a domain that does a certain thing. So domains are basically units of structure and also in essence function of a protein. So remember all the crazy stuff that this thing had to do. So let's see if we can figure out a couple things from it. So first of all, let's start at the left. This is amino acid 1 to 42, and first of all, you can tell that this has 5 domains. P53 has 5 domains: transactivation, proline, DNA binding tetramerization, and a basic domain. And there are other areas here that also do some particular things. So now if we take a look at this, just from our last video, we see that there's the N domain, the core, and then the C domain. And so starting here, we have a transactivation domain. Well, that basically does exactly what you think it does, which is this would activate other proteins. And specifically, this makes sense that this would interact with, for example, MDM2 to activate another protein. And it also activates with other ones as well. And so that part of the protein serves that function. The proline-rich one, and again, you don't need to know these for like a test. It's just to describe to you how protein areas and domains do different functions. In this one, the protein-rich domain has to do with apoptosis. So if the cell, if it's attempted to recruit other proteins and to fix DNA damage and it can't, then this is basically the big red nuclear button, for an analogy. Obviously, we can tell something else about this, is that this is a DNA binding domain. So this protein literally binds to DNA, and that makes sense because its job is to correct errors in the S phase of mitosis. And so that would make sense that this would have to bind to DNA. And you can also imagine from some of the other presentations I've done that if there are certain mutations, missense mutations, where a different amino acid is put into the polypeptide chain somewhere along here, and it were to change the actual structure of this area, you can imagine it might decrease the DNA binding capability and therefore decrease the function of the protein as a whole. Let me finish up the rest of the domain. So tetramerization domain, that already kind of gives you a hint that this is a quaternary protein, and in fact it is. So as you learned in the last video, you have your primary, secondary, tertiary, and quaternary. P53 is a quaternary protein, which means there's 4 little subunits that are all glued together, and this is basically an area of the protein that has to do with gluing them together. The last one we have is a basic domain, and this domain, as you can see, is on the C-terminal end, and basically this is considered a regulatory domain, and kind of what that means is that this domain seems to be necessary for not only transactivation of other genes, but also function of a couple other parts of the protein as well. So those are the larger domains. And there's also smaller areas of function of this particular protein. This one right here, by the way, is NLS. This is called nuclear localization signal. And this allows the protein to move into the nucleus, which makes sense. This one is the same, except it's an export. So this is a nuclear export signal, and this allows the protein to be exported into places where it might need to go, cytoplasm, etc. So all of the numbers have to do with the actual amino acids and which ones have to do with which domain. So now that you have a fundamental sense of what a domain is, we can now use the idea of domains to compare not only homology, but to find out, hey, which proteins happen to have DNA binding domains or tetramerization or whatever you want to do. And this is where a lot of the work in proteomics is done, because this is how we would need to figure out, well, how do we know that P53 interacts with MDM2? How did we do that? There's many ways that you can do that, but some of the ways would be just to kind of isolate that domain from the rest of the protein, and like a plasmid or whatnot, and do things such as like a yeast 2 hybrid or others type of EMSA assays and whatnot. But that's how we can kind of do this. So I wanted to just give you an overarching, secondary video to explain how alive proteins are, and not as boring sometimes as something that you see that looks like this. So thanks a lot.
Yeah, so that was a very very brief discussion about domains and motives. So till now we are talking about secondary structures in proteins, right? So we have talked about alpha helix, we have talked about beta sheets, beta strands, turn, S, and all like different types of alpha helices. But the question arises, if we can see this simplistic protein in front of us, we can see it is not that simple, right? If a protein is present which has alpha helix or beta sheets like structures, then the whole protein is not functional at the end of the day, right? Only a part of it is functional. If you remember from your biochemistry classes, we have enzymes, right? So there are enzyme active site. So the whole enzyme is not the functional, it's not performing the function at the end of the day. It is that pocket which that bulkier protein has which is doing the function, correct, at the end of the day, which indicates that there is something special about the active site of an enzyme which is not present in whole of the enzyme, and hence if a substrate comes and bind to that protein but not at the active site, the function of the enzyme won't take place, correct? Hence, if you remember, for the enzymatic reaction to take place, there are few conditions to be fulfilled, right? The orientation of the substrate and the enzyme should be proper, so that substrate can actually go inside the or sit on the active site or the pocket which the enzyme has to provide, get the enzyme substrate complex, then that enzyme substrate complex is converted to enzyme and product, correct? So this indicates that there is something special about these active sites in whole of the protein. Though the whole protein is made up of alpha helix, beta sheets, loops and all those things, but still the combination of them somehow is different when it comes to the function of an enzyme. So that specific thing which performs the function and has the ability to fold in itself is called domain. The smallest form of domain which is actually performing the function is called a motif. So we'll see different motifs and domains in the coming classes. So let me take your attention to the protein data bank. So basically, what is a protein data bank? Protein data bank is a repository which contains the information about the proteins for which the structures have been determined. And now we know what are the techniques used for determining the structure. It is X-ray crystallography, NMR, correct? So what do we mean by if we know the structure of a protein? We mean that we know the sequence of the protein, we know the 3D position of all the residues in a protein which makes the protein the tertiary structure and the functional form. So you can clearly see how the protein data bank has grown, right? So it's a 2 years back information. You can clearly see how exponentially and beautifully the deposited structure in the protein data bank are increasing, because that there is a technical advancement also in determining the structure of a protein. Hence you can see that there are so many structures in the protein data bank. So let me give you a brief view of the protein data bank. We'll explore it together. 1 minute, let me share my browser screen with you. Okay, I guess I have to change the browser. No, I can share this one. Yeah, I hope you guys can see my my PDB database screen and my cursor also. Here it is. So this is the home page of PDB, that is Protein Data Bank. You can see that there are almost more than 2 lakh structures which are submitted in this database, and there are computed structure models. We have AlphaFold also. We might take half a class maybe in the coming classes to get the idea about AlphaFold, because AlphaFold is an online tool, is a computational tool which actually helps in predicting the structure of proteins which are not yet experimentally determined, right? So the PDB also incorporates such structures here. Also, AlphaFold has a separate database for themselves for the structures which the tool AlphaFold has determined. Okay, then this is the home page. This is a very straightforward home page. So these are the links for other things which we'll see. And then this happens. This is very interesting. Like, so every month if you go to the home page, there is the molecule of the month. It is generally determined by the people who are managing the PDB, obviously, but it gives you an idea about what is running in the structural biology field, what is the importance of it and all that. So these are certain highlights. These are the recent entries. You will get that what are the newer structures being submitted to the Protein Data Bank, and you'll get a one line information also, like by which technique. For example, this structure of 8YF9. So this is a unique identifier for the proteins which are submitted in the Protein Data Bank. So you can see that this structure is determined by the cryo-EM technique, and you can see that it is a Viron particle, right? It's so interesting to see. Then you can go and explore. This is 9FAC. So you can see the diversity of proteins here itself, right? So if we just put a focus here, so this is the first protein which is available here. You can see it's like a channel transporter which is pretty visible, like helices and sheets, mostly helices, right? Then we have this structured subunit type of a particle structure is there. Then this is the one which is like, it doesn't have any structure yet, it has somehow something, so like some structure can be predicted out of it. Then we have this, it's a beautiful structure of, so you can clearly see the helices here, you can see the beta sheets here. So these are even the color coded, right? So we were talking about domains and motifs. So you can see that this green is one domain, this orange is one domain, this bluish purple is another domain, right? Again, it's a cryo-EM structure. Then this, you cannot see any beta sheets here, right, or beta strands here. So this is predominantly a alpha helix type of structure, and you can see it has 2 subunits. On the first hand, we can see that they are somehow identical. So if you want to go into the details, then we can go into this entry and see what are those things. Same. So you can see the diversity. Look at this beautiful protein. It has beta sheets as separate domains, right? So in this one, there is no beta sheet or beta strand. But here you can see that this is beta sheet, this is beta sheet, and this is alpha helices. So there's a lot of diversity going on in protein, but predominantly they belong to those major secondary structures. See, this is another beautiful structure. The diversity in the protein structure is fascinating, right? So, and now for example, if you want to explore for some protein and you want to understand what is that protein and the information present for that protein, so let's take this entry itself. So if you go to this 8SM1J, so if your protein is not here, obviously your protein won't be here if it is not newly submitted or latest entry. So you can search your PDB ID here, or you can actually paste the sequence of your protein here and it will give you the corresponding PDB entry. So let's say if you want to explore this and see what are the informations available in each entry of a protein. So this is the entry which we clicked on. So the PDB ID is 8S1J. Basically, it's a crystal structure of T-N-ethol oxygenase from Stenotrophomonas maltophilia. I'm sorry if I am mispronouncing it, but so it's a type of oxidoreductase enzyme, right? From which organism it is? It's from this. What was the expression system used for getting the structure of this corresponding structure of this protein was Escherichia coli. So are there any mutations reported? It says no. So it was deposited in February 2024, right? Then these are the authors. This is the corresponding paper entry for this particular structure. Then these are the some basic information about the structure now. So what are the basic information about the structure? You can see that the method which was used for determining this structure was X-ray diffraction. The resolution is 1.83 angstrom, and then these are certain values which basically tells you that how confident you can be that the structure which is present here is very, very accurate. Okay. Then this is again the paper which is associated with the publication of this particular structure. Let me show you a very beautiful thing. If it loads, it will be nice for you guys to see. So this is the structure which we are seeing here, and this is the display window where you can play around with this protein and see how it actually looks in 3D space, right? So this is the sequence which is displayed here. So if, for example, if I am at this, so it will actually, if I am at this place, right? So you can zoom in and focus. You can see which residue is this. You can see all the information is available here, right? But there are better visualization tools are present, and I guess we'll take one class in visualizing the protein. It might be of interest to you. Okay, then so it's a crystal structure. It has how many chains? It has, everything is being displayed, so it has like 2 separate domains, right? And it has 2 chains because A and B, right? So you can actually download all the information on your local system and explore and visualize. So if we want to download the information of this particular structure of a protein, so you can either have a FASTA sequence, right, or PDB format itself. So we generally use FASTA sequence as a starting material for determining any structure. So see, this is the FASTA sequence of this particular protein which we are talking about. I hope it is visible to you guys. Maybe now. Yeah, right? So this is the FASTA sequence. So what is the FASTA sequence? The properties of a FASTA sequence is that it will start with a greater than symbol, then followed by the PDB ID, and then basic information separated by the pipe. Okay, and this is the sequence from N terminal to C terminal. In one line, most likely you have 60 residues in one go. So this is the FASTA format of this particular protein, and we can use this to determine and get the information of all the things, correct? So we might take an example maybe in the next class itself and explore from getting the sequence to visualizing it and understanding the basic properties based upon the structure itself, maybe in the next class. Okay, so in the meantime, if you have any queries, feel free to mail me. And I'm sorry I was not able to return or reply to you guys because I was a bit busy. So I'll surely reply to you all, and all the lectures are being uploaded on the YouTube as well as the Moodle platform simultaneously. Please go through them and let me know if you want any particular topic related to the membrane protein, structural biology or function of protein in the membrane specifically, so that I can prepare well in advance and we can have a discussion over it. So thank you so much.